Yes, indeed. Yes. Well, those two illustrations we've just had are very good. And the punch cards, and I'm actually old enough to just remember punch cards, right? They don't write the code, they carry the code. The
Yes.
same with the castle.
Mm.
The design of that comes from outside the castle. The plastic components don't actually make the information needed to put it together, even though it does go together. So, when we look at genes, genes are information, but the information has to be carried on some sort of substance. Uh, and it is very clearly designed not to evolve when you look at how it works. And the interesting thing is the secular scientists do actually recognize that life is made out of information. And every now and again, they give themselves away. This was from an article in the Proceedings of the National Academy of Sciences. All known life on Earth is based on biological polymers. Now, that just means long strings of long molecular strings. So, long strings of small molecules stuck together to make long molecules, right? And these act as information carriers and catalysts. That's molecules that help other chemical processes carry on in the right place at the right time. Therefore, any theory for the origin of life must address the emergence of such a system. In other words, where does the information come from? Now, an Australian man named Paul Davies put this in a little bit more blunt manner. Being an Australian, we tend to be rather blunt. And he asked this question: how did stupid atoms spontaneously write their own software? And where did the very peculiar form of information needed to get the first cell up and running come from? Because no one has ever seen non-living chemicals spontaneously turn into a cell. The genetic information that we study is information that is already in a cell. And if you look at a cell, it has a store of information inside it called the nucleus. And inside of that, there is about 2 meters of DNA, right? That's an awful lot of chemical information, all very, very neatly bundled up and packaged and kept inside that cell. So, how is the information actually stored? Well, it's stored as small chemicals, right? And these small chemicals are like the letters in the code. Uh, and we can compare it with any sort of language we like. Now, the chemical letters are A, T, G, and C. Now, these are some small molecules named adenine, guanine, cytosine, and thymine. You don't need to know that. We'll just stick to the A's, T's, C's, and G's, and they are just small chemicals. And like the letters in a word, right? The individual letters are quite small. It's not until you put them together that you get any meaningful information. Now, they need to be held together in a structure, and that's how we get the very famous double helix that everyone knows about. And there's a very interesting way that they fit together. Um, they fit together by a process that's called base pairing. Now, that the individual letters, that those molecules are called bases. So a DNA molecule consists of two sort of long strands of molecules called deoxyribose nucleic acid, which is a sugar and a phosphate, and that forms the backbone of it, the two strands. And then the bases project into the center between the two long strands, and they form a weak chemical bond, but it's enough to hold it all together. But it can be broken quite easily, and that's important when it comes to actually copying it. Now, there's a very interesting way that they fit together. The A's and the T's all fit together and the C's and the G's. So if we have our 4 letters like this along one strand, right, A will always pair with T. So if you have an A on one strand, the opposite strand will have a T. Right? If you have a T, it will always pair up with an A on the other strand. And the same goes for the C's and the G's. They will all link up together. And the reason for that is that is how the information can be copied accurately. Now, every time a cell divides, the information in your DNA has to be copied, and it must be copied accurately. And because of this base pairing, that's how that can be done. And the way this works is that if you have your 2 parallel strings of information with the base pairing, right, A's and T's, C's and G's, right, you can then split apart the 2 strands and make a new copy. And so we now have 2 identical copies using one half or one strand as the template for the other. Now that happens every time one of your cells divides, and that's going on sort of millions and millions of times all the time in your body. And it's essential that that information is copied accurately, because otherwise your cells won't work. So let's go back to the cell. Well, that's the information that's stored in the nucleus, but in order to be used, it has to be somehow transported. The information has to be transported out into the machinery, into the working part of the cell. Now, DNA stays in the nucleus. It's important that it is kept there. It's like an archive of information, and it needs to be protected. But in order to use it, the information needs to be sent out into the cell. Now, it's a bit like if you imagine a building site, you don't take the architect's original drawings out to the building site, right? You take copies. And this is what happens inside a cell. Now, this is a very simple way this happens. So DNA is an archive of information that is packaged up and stored. If you need that information, you make a working copy. And that is the molecule that Glenn referred to, RNA, and it is a working copy. So it's the same information, but it's not exactly the same substance. In fact, it's only a single strand. And that information then has to be sent to a protein-making machine out in the cell, which is called a ribosome. So the RNA is sent to the ribosome, which grabs hold of it, and it will literally read the information, that series of chemical letters, and use that to make a protein. Now, proteins are the stuff of life. Actually, they form the structure and the function of the body. So this is how we go from DNA information to a physical cell and a physical body, is basically by this process. Now then, there are millions of other chemical processes that then happen around that, but we'll just stick to this first. So what happens with the RNA information? It is a similar molecule to DNA, but not exactly the same. Now, in fact, it is made by base pairing as well. That's how the information gets transferred, right? A bit like copying the DNA, but instead of having T's, right, chemical letters called T's, it has a chemical letter called uracil, so we'll call that U, and A is paired with U. So in order to transfer information from DNA to RNA, we have a similar process, and we end up with that same information, but just slightly different letters. Now, we have transferred that information to different chemicals, but it's still the same information. And that's important to understand the difference between information and this substance that actually carries it, because the substance didn't make the information, it is carried on it. So we now have information being transferred. Each step involves different molecules, but the information is the same. It doesn't come from the chemical properties of the molecules. It's actually in the sequence of molecules, just like the letters in a language. In order to make a word, right, you can have 26 letters, but they can be put into all sorts of different combinations to make hundreds of thousands of words. I'm not quite sure how many words the English language has, but I think it's in the hundreds of thousands, certainly in the tens of thousands. So we are talking about something that behaves like a code. So hence the analogy that Craig and Glenn have used. We are talking about a code. We're talking about information where the components that carry it didn't generate it. They just carry it. But we have an interesting problem with DNA code and RNA code, right? Body structure and function requires proteins, which are then used to make all sorts of other molecules, as well as being the body structure, right? And proteins are made up of small molecules called amino acids. So a lot of biological molecules are small molecules strung together to make bigger ones, just like the components in Craig's castle. You've got a whole lot of small components that can be assembled in different configurations to make a different kind of castle. Now, there are 20 different amino acids, so you can get all sorts of amazingly varied shapes and sizes of proteins. And the information that's carried on DNA and RNA actually codes 4 amino acids. But the interesting thing is we've just said that there's only 4 DNA letters or 4 RNA letters, but somehow we've got a code for 20 different amino acids. So how is that done? Well, there is a solution, and to help us understand that, let's look at code that's carried in English language, in plain language. All right. Now, what does that say? What does that say? Any of you know?
The cat ran off the mat.
Right. Yeah. Yes, you're right. Now, how did you work that out?
I suppose I already knew the letters, the words within that.
That's right. Yes. You recognized combinations of letters. It's not just a random lot of letters. You looked at that and you said, yes, there are particular combinations that go together. And what you've done is you've separated out the combinations, the 3-letter combinations, using spaces, right? And we go from what could be just a random collection of letters that's been thrown together into a meaningful sentence. Now, what you've done there is apply what's called a reading frame, right? We could keep all of those letters together and just split them up with lines, right? In written language, we use spaces as a reading frame. Now, in DNA and RNA letters, right, they're all stuck together. They don't have spaces together. But in order to make that information meaningful, it does have to be read in groups. Now, they happen to be groups of 3 in English. Of course, we can have groups of 2, 3, 4, 17, however many you like. Now, here's an interesting problem. What happens if you change the reading frame? Right. So if we were to just move it a little bit along, now, to make it easier, I've split it up again using spaces as the reading frame. Now, have we improved that information?
Well, it becomes meaningless in English.
Right. Yeah. Yes, that's right. We've garbled it. And so what we've got here now is another extra layer of information. Not only is the code with all of the letters neatly written out, we've got another layer of information. In other words, the cell has to know that machinery, that ribosome machine, has to know where to apply the reading frame. In other words, where to start reading the sequence and in what order. So the meaning is in how the code sequence is read. So again, we've got a property of a mind that's outside of the substance that's being used to convey the information. Now that adds an extra layer of complication. So you saw with Glenn's slide there of how many different combinations you can have of just 4 letters. Well, just imagine how much more complicated it is when you have to read them in groups of 3, but you have to get the right groups of 3. And because they're all stuck together in one long sequence, you have to start in the right place. Otherwise, the whole thing is rendered useless. So let's go back to RNA and protein. So here was our problem. We have 4 bases, but 20 amino acids. Our solution, 3 bases, will code for one amino acid. So we have to apply a reading frame, and that will tell us what amino acids that need to be strung together. So those words at the bottom there are actually just a shorthand for the names of amino acids if we apply the reading frame as it is there. Now imagine what would happen if the reading frame was just shifted. You'd end up with different amino acids, which means you might end up with a different protein. And most of the time, you would end up with a useless protein because the structure and the function of a protein depends on getting the right sequence of amino acids in the right order. So randomly changing DNA letters is not going to help. In fact, it's going to muddle things, just like my little sentence about the cat running off the mat. Right. Now, when cells are constantly dividing, the information has to be copied accurately, and the same with the reading frame has to be applied accurately. Now, when you're copying information, you think about that. It's quite easy to get typographical errors. Now, typographical errors do not add useful information. In fact, they degrade it. So the wrong amino acids, if we're reading our sequence wrongly, or some of those letters have been transposed or left out or knocked out, you end up usually with a useless protein. So it is very important that information is accurately copied each time a cell divides. And if you look at the process that's involved in that, it is hideously complicated. Here is a simplified version, and I can assure you this is a simplified version. The number of molecules that's involved in copying information, making those 2 complementary strands come apart and get copied into 2 accurate copies is very complicated. And if we just have a closer look at the first part of that, during that process, the information is actually copied, but then it is also checked and edited. So it's as if you've got a proofreader over your shoulder watching what you're doing if you are copying information. Now that is really brilliant. So we have a built-in system for copying, proofreading, editing any mistakes that are found straight away during the copying process. Now, even after the information has been copied accurately, it can get damaged by various cellular processes that happen or various things that come in from the outside. And so your DNA is being constantly surveyed to make sure that it is remaining intact. And there are built-in repair processes. So there's constant surveillance of your DNA. This is going on all the time in your millions and millions and billions of cells in your body. And any mismatched or damaged DNA can be repaired. And again, you have to have, this is a very, very simple explanation of it here with these diagrams. There are dozens of molecules, and they're huge, complicated molecules as well, involved in this surveillance. And if a damaged piece of DNA is found, molecules will come along, the repair gang, as it were, will come along, take out the damaged or mismatched bits, and and put in new bits using that base pairing as the template. Now, sometimes the DNA becomes just hopelessly damaged and it can't be repaired, which means the cell effectively becomes non-functional if the DNA is too badly damaged. And there is actually a nuclear option, as it were. If that cell's DNA is too badly damaged, the whole cell will be dismantled. And that is a process that's called apoptosis, which just literally means falling over. But if the DNA is irreparable, a whole cell is dismantled and will be replaced from a clean copy, as it were, from elsewhere. And what happens is that the cell literally starts splitting itself up, and it will shrink and shrivel up and become fragmented. And so that way our cells, the integrity of our cells is maintained most of the time. Now, some diseases, particularly some cancers and some other diseases, result of this repair process being knocked out. In other words, the surveillance and repair mechanisms get knocked out, and that's where we get a lot of diseases from. But healthy tissue is being constantly surveyed and repaired. So let's summarize what we see in the way genes work. Right, we have layers of information. It's not just lots of information. We've actually got layers of information in terms of how the DNA and RNA letters are put together in sequence and how they are read, which means that has to be built into the cellular machinery that reads it. Information is a product of a mind. It's not the product of matter and energy. All those C's, T's, G's, etc., they didn't make that information. It's the way they are organized, and they can't organize themselves. Once they are put in the right place, they will stay there because of the chemical properties, but they won't put themselves in the right place. Now, when the information is being copied, there is a copy, check, edit system that will maintain the accuracy of the DNA in cells and maintain the accuracy of the RNA. And there is ongoing surveillance and repair for most of the time. It's loss of that surveillance and repair that results in genetic diseases. That's not evolution, that is devolution if you lose that repair process. So healthy cells are designed not to evolve. DNA is not designed for evolution. In fact, it is designed to stay the same. Now, as Glenn mentioned, in the Bible, we are told 10 times that God made things to reproduce after their kind. And that is built into DNA. DNA is designed for after their kind. It's masses of information set into a whole lot of complicated cellular machinery which keeps it the same. It's designed for after their kind. And I think we've got some nice examples of that to look at. I think Craig's got one as well. So what do we see out in the real world? Well, we don't go around with a microscope looking at DNA, but we do see the results of DNA being designed for after their kind, for things to multiply after their kind.
Doesn't the 98 to 99% similarity between human and chimp DNA prove evolution? Diane, perhaps you'd like to kick that one off, and then we'll go around to some comments from the others.
Well, 98 to 99% of what? This is where understanding that DNA is information is important. Very small changes in the letters of words can result in huge changes in the actual meaning. So simply because you have a very similar number of A's, T's, D's, and C's, even in similar sequences, does not mean that you are producing exactly the same results. Just like very small changes in words can result in quite spectacular differences in meanings. I mean, that's where typographical errors can be extremely funny sometimes. That's no longer being used as a comment these days, because people are starting to understand the importance of information. The original 98.4, I think, comes from a book that was written before the human genome and the chimp genome were actually sequenced, and it was worked out from just getting strands of human DNA and chimp DNA and seeing how well they fitted together. And sometimes they do and sometimes they don't. But if we go back to our analogy of building things using information, if you were to write out all the instructions for building two buildings, sort of, a brick shed and a castle, well, a lot of the components will be the same. Right? They might be both made out of bricks. They might both have windows in them and things like that. So if you were to write out all the information on how do you build a window or a door, or how do you make bricks, that information will be the same. But the results are very different because of the control. In other words, where do you put the windows? Or where do you put the doors or the roof and things like that? Now, the amount of information that's needed to say, put this window here or put it 5 feet away is, if you wrote that out in words, it would be very similar words, but the results would be very different. So simply because we have a lot of the same information just shows that our cells are made out of a lot of the same stuff. Well, in fact, if you looked at the information there is in, say, something like a plant, there's a lot of information there, which is the same as the information in a human body, because there are certain things that all living cells need to have. But that doesn't mean you're 50% of a plant. And so you've got to think, that's why I said, 98% of what are you actually describing? And when you get back down to looking at how information works, it's not quite as simple as, well, yes, we've got all these letters that are same, and we've got 98% of these letters that are the same. How are they organized? And what are the letters actually telling you to do?
Well, correct me if I'm wrong. Correct me if I'm wrong, Diane. But doesn't that statistic exclude junk DNA? And it's now only at like 70% or something like that, and the junk DNA now is not recognised as junk DNA.
As
it
junk,
excludes
that's
more
right.
than that. They only compared the 2.4 billion letters that actually matched. They went through and selected only the sections that matched. And so they only compared the DNA that matched. They excluded 1.3 billion letters that they knew did not match. So the question really is, why didn't they get 100% match? They didn't get 100% because they weren't perfect in their selecting of what strands they were going to compare. So it was a total fraud, and that's why they don't use it anymore. But it's still being promoted.
Yeah. So
yes, there is an interesting study that was done quite a few years ago. Genes are actually packaged into what are called chromosomes, which are just long strings of genes. And so someone thought, well, it'd be a good idea if we compared the chromosomes, one chromosome in a human cell and one chromosome in a chimp cell. And they did it with two very small ones, lined them all up. But then, as I did in my little slideshow there, you've not only got to look at the information, but you've got to look at the outcome. What is that information coding for? And very small changes will make quite big differences in the amino acid sequence that is being coded for. And so they did a study where they looked at a very small chromosome, human example, chimp example, sequenced it, and worked out what results would you get? What proteins would you get out of this chromosome? And there was an enormous difference. It was about 82% different in terms of the outcome. And that's what matters. Now, we did write that up in our newsletter. It was quite a few years ago. So if you go to our fact file and just look up chimp genes, you'll find that report. I can't remember the exact reference off the top of my head, but go to our fact file. It is described there.
And that's why it is very similar to computer programming, because I can tell you, if you got one little dot off in my codes in those punch cards, it's totally different and it's totally wrong.
Yeah, well, you can use statistics in any way, can't you? Like 90% of children like to climb trees, so they must be little monkeys.
I think you've hit on the nail on the head there. Statistics is it's not the father of lies, that's the devil, but it's certainly the offspring of the father of lies. And this is the point that John made. If you've seen the interview that John had with the BBC, the one that got him banned from the BBC, and it's an amazing interview. It's up on our YouTube channel. Go and check it out. But the interview actually brought up this very point, right? Surely, the fact that we're 98% similar in DNA to a chimpanzee shows that we must be related to them. Well, aside from the point, as we've just discussed, that 98% statistic is effectively false, provably false, because they ignored a huge amount of the DNA in it. Let's just assume for the second that we are 98% similar, right? Now, you know that there is some difference between us and a chimpanzee, because there isn't a chimpanzee sitting on our panel tonight, right? The fact that we are able to converse, the fact that we've built up a civilization, the fact that we're able to write books and cook dinner and all this kind of stuff shows that there must be a significant difference in reality between us and chimpanzees. Now, that actually, even if we are 98% similar, shows an enormous amount of design that has gone into the genome to be able to make such completely different creatures that are 98% similar. So even if we are 90% similar, we're still evidence of amazing design. And you can go one step further and link it into the way that we've been working with codes, right? Glenn's spoken about codes. Glenn has spoken about codes. Craig has spoken about building castles. Diane's spoken about the design of the DNA and the like, right? And the example which John used is very good. You can take a statement, much like we had a statement with Diane earlier, right? With the cat, and you muddle it up. Well, you can go one step further, because you can get the statement, God is now here. You can make one deletion of a space. You can have 100% of letters in 100% the same order. You delete one space and you get God is nowhere. And that's a 100% opposite meaning of God is now here. And it hasn't even changed any of the order. You've just deleted one space. It's a tiny fraction of a change, and you can create an entirely different meaning to your code. And then really, if we are 98% similar, the final question that you should ask is, compared to what? Because just one deletion, if it's only a sentence long, 98% is quite significant. But we now know that there are, 3.1, 3.2 billion base pairs in our genetic code, right? That's an enormous amount of difference in just 2% all of a sudden. And so this whole 98% similar argument, it really just doesn't hold any water, even if we are 98% similar, which we're not, and we've just discussed that. So there's a whole load of stuff that we could delve into here. But Diane, just remind us, where can people go if they want to find out more about the fact file and these kind of reports?
You can go to our main website and just go to what's called the fact file, or you can go to it directly, creationfactfile.com.
All right, great stuff. Thank you.
If you want to get them before they get onto the fact file, you get our newsletter and you'll get them. Straight away. So the fact file is an archive of our newsletter. Now, when you look up the fact file, you don't need to know when they were originally published in our newsletter. You can just do a normal keyword search and find them.